Yes, good afternoon and all. Yes, yesterday as sir discussed, or today morning session he has discussed about ML and QSR modeling based on the obtained drugs. Based on the above obtained by the drugs which has taken from that ML and QSR modeling, you are going to where we are in that ML and QSR modeling, you are going to predict like you are going to check how the drugs are effective in what way. Here in further in docking, we are going to check whether those drugs are able to bind to the particular receptor or what. So we can see that in the molecular docking where we can convert, where I am going to discuss about what is molecular docking and what are the types involved in docking and categories of docking, and followed by mechanism involved in binding, and followed by scoring and its evaluation of scoring function. Let us see docking. What is meant by docking? Basically, in a basic definition, we can say that docking is where the ligand is going to bind a particular receptor. In what extent the ligand is going to bind to the particular receptor, that's called as a docking. Where docking is the identification of low energy binding modes of a small molecules within the active site of a macromolecules or a receptor, and whose structure is known. We are going to identify the in what way the ligand is going to bind to that particular receptor. Docking is the computational determination of binding affinity between the molecules, like you can see protein in between the protein structure and ligands, or it in between the protein, or in between the protein and the DNA. Next, the given protein has a ligand, find out the binding free energy of the complex formed by the docking term. Where we can see here the ligand and receptor, you can show here where in what extent that ligand is going to bind to that particular receptor. We are going to find out, or we are going to look into it, and it is called as a docking, where the softwares utilized here AutoDock Vina, Schrodinger suit, Stardrop and Gold. Where AutoDock and AutoDock Vina are the freeware tools, you can freely accessible, where but in Schrodinger and Stardrops are the licensed version, they are commercial software. Next, we will look into the types of docking. What are the available types of docking? We can see here. One is rigid docking and is flexible docking. Rigid docking, where it involves both receptor and ligand are in rigid conditions. Like it implies like lock and key model, where both ligand and like lock and key models means like it's both receptor and ligand are in rigid in nature. The conformation will not change over there. As a method is like for a particular key or a particular lock, a particular key only you are going to utilize to open that or work on it. So like that way here, the particular protein you're going to fit into that particular ligand. Based on that, the rigid docking has been formed. Where in flexible docking, both ligand and one of the molecules like ligand or a protein is get flexible, where we can use here the ligand is got flexible rather than the protein. The protein will be in the rigid in nature, where ligand will be in the conformational changes. It can change their conformation to the binding pocket. Like you can see over in the image, the ligand is getting changed its conformational sites to binding to that particular binding site. Followed by basic binding mechanisms involved here. How the basic mechanisms which involves in the binding, the complementaries between the ligand and the binding sites. You can see two of them. It is steric complementaries and the physicochemical complementaries. Steric complementaries, the shape of the ligand is minimized in the shape of the binding site. As like explained in the lock and key model, it should have particular interactions. If the ligand has a active site, it has to bind to that particular receptor, means it should have it is giving a hydrogen bond donor, and based on that receptor should also have the hydrogen bond acceptor. Based on that coagulation or complementaries, it is going to work out on it here. The mechanism is working on. Next, physicochemical complementaries, like you can see PK and other things, etc. Based on that, these are the complementaries we can see over in the binding mechanism. Followed by categories of docking. There are two categories we can see over here. One is protein-protein docking and protein-ligand docking. In protein-protein docking, both molecules are rigid, where you can see like lock and key models. There is no orientation change in the or conformational changes takes place in both protein and protein. One is modeled on, one is will taken from other PDB or databases. So both where both the proteins are rigid in nature, there is no conformational changes we can see over. It will going to bind at the surface of the protein only, not into the active pocket. Where in protein ligand docking, we can see the ligand is flexible, not the protein. Where the ligand will changes its conformational to bind to that particular receptor. Based on the conformational changes, we are going to based on that conformational changes, for each poses we will having on score. Based on that score range, we are going to order the docking score and scoring functions. Next, followed by what is docking and scoring. Like we can see scoring like to the place of ligand into binding site of a receptor in a manner appropriate for a optimal interaction with a receptor. To evaluate a ligand receptor interaction in a way that may discriminate the experimentally observed made from the others and estimated through binding affinity. We can see here the ligand is where ligand and receptors are talking together, that is binding to each other. In between you can see over, it will changes the conformational changes. Based on the conformational changes, the ligand will going to bind to the particular receptor. Based on this, which is having a optimized high activity, high scoring or high value, high energy, it will be scored as ranked as a top molecule. Based on that, we are going to see the scoring functions. What are the components of docking? Here, the typically protein ligand docking software consists of two main components which work together like search algorithms and scoring functions. In search algorithms, as I mentioned, where the ligands will change the conformations. So to bind to that particular docking, the particular receptor, it will go on changing the conformation. Based on that conformationals changing, the docking score is the scoring function is ordered. Based on that scoring function, the docking score is ordered. That each docking score is calculated using the scoring function or a binding affinity for a particular pose, and that binding affinity or a scope of representing in the strength of the binding affinity, where the based on that particular scoring function and what is the energy it is having that ligand is going to bind, it will have so much of conformation. Where we can see if we docked means it will show the so much of conformations, and in that result analysis which is explained by Sunil, what is the result analysis and how it will shows the conformations, and in that conformations which is having more energy, that will be ranked by using scoring functions. Next, further the scoring functions also depends on the both the ligand and protein interactions, where it involves hydrogen bonding, electrostatics, van der Waals and hydrophobic and pi interactions. These are the interactions which falls under the scoring functions. Based on this only the scoring functions, and we can rank the compounds which is binding to. Based on this ranking, we are going to rank that, or we are going to analyze the result. Based on this, we are going to evaluate the scoring functions. Then also these scoring functions are applied in the wave equation. Based on that wave equations, it will give the proper values, and at last we will get to the what is the minimum or binding affinity we are going to get through this. We are going to evaluate. Next, further the demo is given by Sunil. ಮಾಡಿಲ್ಲ ಎಲ್ಲ ಇನ್ಸ್ಟಾಲ್ ಆಗೈತೆ ಎಲ್ಲ ಇದರಲ್ಲಿ ಆಮೇಲೆ
Good afternoon. Shall we start the demo? I think already you people know how to use Discovery Studio and PyRx, right? Before going to docking study, we have to create one folder in which we have to save all the files regarding docking. Right now, I am saving the file as a demo. right? This is the workspace of our today's session. First step is to download the PDB format, PDB ID for hypothesis, whatever you created or some diseases. Second one is you have to download the ligand and you have to make the cluster. Afterwards, third step is docking study for both the ligand along with target. Please, I created an hypothesis that is I am working on Alzheimer's disease, that too GSK3 beta as a target. I am focusing on GSK3 beta target. For that, I have to download the PDB format, right? Just if you click on PDB file, just open that. This is the open. Once if you open the PDB file, just here type your target name. Like for example, I am typing as GSK3 beta. Here you can see that almost 23 it is there, but we don't know which we have to select. As session 3 told by Basanagowda, that is how to select a protein from PDB data bank, right? Some of the criteria is there, right? You can see here options like that is scientific name of source organism. Right now we are working on Homo sapiens, right? That's why you have to click on Homo sapiens. If you are working on bacteria, you have to select bacteria first. E. coli is there, means you have to select E. coli and all. Again, experimental method. Different type of experimental methods also is there. Right now only one it is there for GSK3 beta, that is X-ray diffractometry. Just click on that one. You need protein or DNA or enzyme, anything if you require for specific diseases, right? Right now I require the protein. That's why I'm selecting as protein. If you require to work on DNA means that time you have to select DNA. If you want to work on enzyme means you have to click on enzyme. Right now I'm going to work on protein. That's why I'm selecting as a protein. That resolution, it should be on lesser than 2.5. As yesterday session has explained, right? If lesser than 2.5, it is good quality. That's why I am selecting as 1.5 to 2. Again 2 to 2.5. Again we need latest version, right? That is why I am selecting 2020 to 2024. At last, if you run this one, again it will shortlisted. Now you can see this. Out of 23, you will get 4. By easily from 4, you can easily download anyone, right? By observing that one. See here, first one that is 2020. Again, second one is 2022, 23. Again, third one is 2023. Fourth one is 2023. But last 3 are 2023. Out of 4, again you have removed first one. Again you have only 3. In that again you have to see for the classification if there is a mutation or not. If mutation is there means that also cancer. Lastly you will get 2. Again you have to see in that latest version that you have to download. Again if you want further you have to see resolution also. See here, for first one it is having 2.14 resolution. For second one that is 2.33. For third one it is 2.09. For fourth one it is 1.85. In this out of 4, it is fourth one having 1.85, right? That's why I'm going to select fourth one. Just click on that one. See here, some of the criteria, that is Homo sapien. There is no any mutation. Even more, it is less resolution. Even more, it is latest version 2023, right? That's why I'm selecting this as a PDB ID for GSK3 beta. Just go here, download file, and you have to download in PDB format. Just click on that. You can see that PDB format, right? This is the first step. After downloading the PDB, you have to clean that protein, right? For that, you have to use Swiss PDB Viewer. That is the tool software to clean that protein, or else if any amino acid is missing in this protein, you have to refill, or else to remove the water molecule which is present in this protein, that also you can done by Swiss PDB Viewer. If you open this Swiss PDB Viewer, this is the workspace for that. Just drag your protein in that Swiss PDB Viewer. Just click that one, that. It is asking, okay, just click. This is your protein. Now you can see that how many amino acids are present in that protein. What are any heteroatoms are present is there or not in that protein. Any water molecule is there or not. If any amino acid missing or not. For that we have to use this tool, that is Swiss PDB Viewer. Just click on Wind Control Panel. You can see here. These are the amino acids which is present in that folder. Here you can see that purple, right? It means it is reloaded that valine amino acid. It means in this protein, valine, it is a missing amino acid. It is reloaded, that one. Automatically it will reload that one based on that protein structure. Again, you can see that how many chains are present. Right now it is A chain also is there. Again, B chain is there. For that, you have to go again PDB. Downside, you can see A chain, which is having 351 amino acids, and B chain, which is having 17 amino acids. Again, you can see here co-crystal. It means here chain which is C, which is similar to A. It means that co-crystal is binded to A chain. By using this co-crystal, we will identify the active region of that protein, that is XYZ coordinates. To determine that XYZ coordinates, we will use this co-crystal, and we have to calculate the XYZ co-crystal for the activity of that region for protein. Now we require only A chain, right? That's why I am removing the B chain. Just select this one. I selected all the B chains. You can see it, right? Just click on B chain and up to end. That is totally it is having 17 amino acid. Just click on that one. Afterwards, you have to go for build. Here you can see the remove selected residues. Now I removed the B chain from this protein. Now it is containing only A chain along with one co-crystal, that is 6VL401. That is the co-crystal. It means by using that one, we will identify the XYZ coordinates. Now this protein you have to save. File, save as current layer. We have created one folder, right? In that only you have to save. This is clear protein. I am typing as clear protein or target. While typing clear, again, don't give any space. Instead of space, you can use the hyphen or any symbol, because this will run all over Python script. That time space won't detect in Python script. That's why I'm using some space, hyphen. You require in PDB file, PDB format, right? That's why I'm selecting PDB file format and save. Okay, this is now ready. Now for docking, we don't require even that co-crystal also. That's why you have to select that co-crystal. Same thing, build, remove that heteroatom. Build, remove. Now save. Again you have to save. I am saving as prepared target. If you open that file, you can see, observe, clear target, that is protein along with co-crystal, and clear target, that is containing protein along with co-crystal, and prepared target, that is only protein. It clear target is the right, that is used, I am using that for identify the XYZ coordinates, and whatever prepared target file is the right, that I am using for docking studies. Now protein preparation also done, protein amino acid and water molecule everything done. Next step is ligand downloading. For that, I am going to use some of the examples for Alzheimer's disease. We know that, right? Standards already, donidibab, right? Galantamine. For that, if you want to download, you have to go for PubChem. Open PubChem. Just type donepezil. This is the standard for Alzheimer's disease, right? That's why I'm taking as donepezil. Just enter. You can see this one. Afterwards, here you can see the option that is download. Just click on that one. Here you can see that 2D structure as well as 3D conformation. For if 3D conformation is there, means you have to save that file in 3D. If there is no any 3D conformation, you have to save to 2D. Right now it is having 3D conformation, right? Right. That's why I'm going to save in 3D format, 3D conformation only in SDF format. You can see that options right downside, SDF format. Protein has to be saved in PDB format and ligand has to be saved in SDF format. Just click on that one, SDF format. Okay. I'm taking one more example also. That is galantamine. Just click on that one. Now same thing, download. If 3D is there, means you have to go for 3D. If there is no any 3D, you have to go for 2D. If there is no any structure, because sometimes you people have go for synthesis, right? That time that structure it won't be in available in PubChem, right? That time you have to go for ChemDraw, ChemSketch, both is there, right? You have to draw and you have to save that file in SDF format. That also one option is there. Okay. Save. Now I'm creating one more folder, that is raw file. I'm pasting that two ligands. Now we downloaded two ligand slides. You can make 2 or 10 or 100 depending on your system. Right now I am taking only 2 examples. Okay. Here afterwards you have to make that 2 single SDF file into 1 zip file. It means you have to make cluster. It is like zip file. How we are making, right? So many files is there, means we will create one folder and we have to make zip file, right? Like that you have to make these 2 SDF file into single file, that is zip file. We will call it as cluster. For that I am going to use as a tool, that is Discovery Studio. Open Discovery Studio. Just click on new, but new. select all the files, SDF files. Right now it is having 2, right? If you have 10, if you have 100, just select and insert in Bio Discovery Studio. Now you can see it right here. I inserted as 2 files. Just select and drag into that. Here it will give PubChem ID. If you want to change that name, you can change it. That is 3152, that is donepezol, and 9652, that is galantamine. If you want to change, just click on that one, write the name for that. For example, see. If you want to give the name, you can give, or else you can forward. Right? Now, whatever you are seeing, right, empty place, just click on that, Control A. Now you are observing yellow color, right? It means both are selected. Both are selected. Afterwards, you have to make that 2 files in single file. For that, you have to go File, Save As. Here you can see that option that you have to save mole SD file as a cluster. I am typing as cluster. Where you want to save, you have created one folder, right? In that folder only you have to save raw file cluster. As a cluster, it means it is a zip file. It means it containing both the ligand as well as donepezol as well as galantamine. Save. You can see the file. First one is this is donepezol and this is galantamine. This is cluster. It means it containing both the ligand. Now protein has done. Again ligand has done. Third step is docking. For that, I am going to use PyRx as a software. It is a free tool. Before going to docking, you have to see the XYZ coordinates of that protein, right? For that, you have to use one more tool, that is Discovery Studio. Open again Discovery Studio. Here you can see it, right? A clear target which is containing protein as well as co-crystal, that you have to drag in Discovery Studio. This is the protein along with containing co-crystal. Just if you click on this button, you can see the option. Here you can see heteroatom, right? Just click on that one. Just here you can see that down, define and edit binding site. Just click on that one. From current selection, I selected as heteroatom, that is co-crystal, right? That's why I'm clicking on from correct selection. You can see the option here. Here you can observe this pair, right? That is means co-crystal as present in that region. It means it has selected that region where co-crystal as present. From that we have to identify that is the active region of that protein. For that we require coordinates. At x-axis how much is there, y-axis how much is there, z-axis how much is there, we require, right? For that you have to create pair objective. Here you can see the option x, this is y, and this is the z. It means x-axis that is 4.85 and y-axis that is minus 11.75 and z coordinates that is 30.52. Before going to docking, these values you have to note down that one. Right, now done. This is X, Y, Z coordinate. This means in this region, that protein will be active region. The active region present in that protein. In this region, the active region is present of that protein. Now close this one. Now open PyRx. It's taking some time. This is the outlook of PyRx. Before going to that, you have to choose the workspace. After getting docking and all, it is asking where we have to save the result file. For that, you have to go edit, preference. Here you can see the option that is workspace, where you want to save that all the files, whatever you are doing, right, where we want to save those files. For that, that's why I created one demo file in desktop, right? That I am going to choose desktop demo. Okay. Here also okay. Just close this one. Refresh the system. Do one more time. Edit, preference, workspace. Click on that one. Select the demo folder. Okay. Again, okay. Close. Everything you have to close. Just refresh the system. Now you can see that folder. Like before, I didn't download any files, right? I didn't create any files. Only raw file I created, but automatically 3 files are here. After that, you have to do again. Go to open PyRx, edit, preference, select your workspace. Okay. Afterwards, close that PyRx mode. Afterwards, refresh. Again, you have to see these 3 files are created or not. Up to this one, you have to do repeatedly. Because that system, if your system fast it is there, means it will create one time. If your system very slow, means we have to do one more time. Okay. It means whatever Python or anything, docking result, everything will be saved on this folders only. Again, we have to open PyRx. Open file, load molecule. Select your folder. You can see that load molecule. It means you have to load your target. Whatever you created a prepared target, just click on that one. Now you can see that this is your prepared target. There is no any co-crystal or heteroatoms. Pure protein that is. Just click on that. Right mouse click, AutoDock, make macromolecule. It means whatever we have incorporated PDB file, right? That is converted into PDB Qt format. Because for doing docking, we require PDB Qt format. That is why I'm going to create PDB to PDB Qt format by clicking that right mouse click, AutoDock, make macromolecule. Afterwards, you have to choose again file, again import. Now you have to import your cluster. It means your ligands. For that, you have to click chemical table, next, choose the demo file. You have saved in raw file, right? Open raw file. You can see the cluster. Just open that one. Downside, you can see it. How many ligands are present in that cluster? We have downloaded only two files, two SDF files, right? That's why two files. If you have 100 compounds, you can see the 100 compounds. If you have 10, you can see the 10. Right now it is 2, right? That's why 2. Again, you have to convert that SDF file to PDB-QT format, right? That's why I'm going to click on that one. Just click, right mouse click, minimize all. It means that structures will get minimized. How we are separately doing by Avogadro's and all, right? But in this already it is there inbuilt. Minimize all. Again, you have to create, convert all to AutoDock Ligand. That is PDB-QT format. It means what and all we download a SDF file, that is converted into PDB-QT format. Just click on that. Now you can see that it is converted as PDB-QT form. Now target also it is in PDB-QT form. Again, ligand also it is in PDB-QT form. Afterwards, docking. Just click on AutoDock. Afterwards, click ligand. Select all the ligands along with protein. Just click on Vina wizard. Start. Again, forward. Here you can see this one. This is the grid box. It means we have created X, Y, Z coordinates, right? It means we have to identify this protein, which is the active region, by adjusting the X, Y, Z coordinates. Right now it is having 1.8, minus 1.7, and Z is 30.1. If we click on maximum, it will select all the protein. That time it will go induced fit theory. Have you studied, right, in biochemistry? Key-lock model and induced fit theory. It means induced fit theory means we will select all protein, but our drug will go and bind wherever it requires. But in key-lock model, we will set the range, that is X, Y, Z coordinates. It means our ligand will go and bind in that region only. That's why we have to select X, Y, Z coordinates by adjusting this box. Okay. We have, that is X coordinates is 4.85. Now you can see 4.1, approximately it is there. We require 4.8, right? Now you can see it 4.8 X. Now Y we require minus 11.7. Minus 11.75. Now we require Z, right? Z is 30.52. Right now it is, we require 30.55. Right now it is 30.5. It is okay. Now forward. Here you can see docking as process. This is the docking. You can see it. Don't it is running. After running, you can see the observe of code. Now docking is done. Now you can observe this code. If you want to download that source, here you can see second option, that is CSV file. Just click on that one. Choose your file. Result of docking. I am typing as result. Molecular docking. This one close. You can see that file. Now you can see Doni PSL, right? It is having 10 different structures. But here, once if you consider RMSD, where RMSD 0, which defines means that protein along with ligand, there is no any other interaction. If you get any RMSD value, it means that protein has binded to that protein, but some other interactions has act on that protein. That's why we will not consider that one. Just delete those compounds. Control Y. Now you can see. Now you can see donepezil having binding score that is minus 7.6. Not only consider docking score, we have to see the amino acid. What kind of amino acid is present? It means binding with that protein. Your ligand, what kind of amino acid has binded to that ligand protein? For that, you have to use tool. You have to make that complex by using PyMol. Afterwards, you have to do 2D and 3D of that structure by using Discovery Studio. For making complex, you have to use PyMol tool. PyMol This is the PyMol. This is a workspace for that. After getting result, you can open that macromolecules file. Again, prepare target. You can see the different files. This is configuration. What we kept XYZ coordinate that is related to that file and donepezil and along with target. Just select both. Just drag this one. Now you can see that one. Protein along with donepezil. You have to make complex. It means some of the softwares it will give automatically complex, but PyRx it won't give complex automatically. We have to make complex by using PyMol. For that, you have to go file, export molecule, again multi-file. It means we have two files, right? One is prepare target along with drug, right? That's why I'm selecting multi-file, and we have making as single file, right? That is, I'm selecting as one single file. Save as a complex. Desktop demo complex. I'm saving as complex. As PDB format. I'm saving as complex is in PDB format. Save. Now close this one. You can see here complex file. It is in PDB format. If you want to that 2D and 3D structure, we have to use Discovery Studio again. Click on new. Just select your complex and drag this complex. You can see here. This is your docking complex. It means ligand and drug has to be docked. Now, if you want 2D and 3D structure, just click here, define receptor. Again, click on OK. Here you can see that option. Here, define receptor, ligand interaction, it is showing where it is ligand present and show 2D diagram. Now you can see that part. This is the 2D of that protein. It means this is the structure and these are the amino acids of that protein. It means here you can see what kind of interaction it is there. Whatever you are seeing, that is alkyl interaction and pi alkyl interaction. This is the 2D structure of that ligand complex. If you want 3D, go script, visualization, do publication quality. It means we have to create one quality. Afterwards, you have to go structure, label, add residues, structure, surface. We have to give the color in different manners. For protein one color and ligand one color, right? For that, right now I am giving color to that protein. For that, you have to go structure, label, and you have to give which color you want required. You have to give which, again, if you want transparency, you have to adjust by adjusting this one, and opalescence also. Again, click OK. Now you can see that. Okay. Now I gave the structure, I gave the color for structure. That is protein. You can see this is the protein. Inside your ligand will be there. If you zoom in, you can observe that one. Go inside, you can see that. You have to use mouse during adjustment. Okay. Structure. Okay. If you want to give the name for amino acid, which amino acid has interacted means, for that you have to go structure, label, add. You require atom or residue. Amino acid will be residue, right? That's why I am selecting as residue. In which format you require? 3 letters with along with where it present IDs and all, right? That's why I am selecting as 3 letters and ID along with insertion code. Just which color you want to give. Just click opposite to green color. I'm giving as red. Just click. Again, if you want to style here times New Roman, you require Arial or font size, how much you require. Just apply. Okay. Now you can see it, right? These are the amino acids. If you zoom in, you will get to know that. This is the 3D structure of your complex. If you save this file and you have to paste in our publication, they will accept in Discovery Studio also. If you have any doubts,
instead of switching to many softwares, is there any one software we can do all the things like visualization, docking,
if you know
site
Python,
interpretation?
yes sir. If you know Python means you have to write Python script in single file. Just by clicking, you can do everything. Even if you have to do PDF to PDBQT, SDF file to SDF PDBQT, and even more docking along with complexes also.
By coding.
Yeah.
Instead of coding, there is no any softwares like that.
Right now,
No,
no.
you can go with the paid versions. Like if you make the complex,
Coding.
right, you can use Master Viewer. That's coding just Master Viewer is an academic freeware where you can able to visualize like a Discovery Studio. So, and rather than the Spimal or what we call, we can use Chimera, we can use VMD for visualizations and all. But it is easy, user friendly. And for the students, we have to
Free
look
overall.
into. So that's the thing.
Thank you.
Any other questions?
Sir, actually one doubt regarding that protein selection.
Protein
selection.
selection. Okay.
Okay. So you told that we can select a protein. That is the latest protein we can select. But when we are going through the literatures, suppose if we are selecting a latest protein, we can see only 1 or 2 literatures. Okay. So whether we have to select that protein or select a protein with more reference.
That also we can choose. Sir. It's what do we call? We need to get a best one. So in case if they have done and the results are proper, while we carry out. If we get the right answer, means example to cross validate some question. What do we reviewers are asking? You said that you have done a document. How do you validate that this is the packet, whether this is the thing? So that time we can able to justify this paper, this score came for us, this score came. So we have done. That is up to us how we are going to justify the reviewers comment. So you can use either the literature review or through this method. Both the methods are acceptable, sir.
Okay. And one more doubt regarding that protein selection. Actually, normally we will select Homo sapiens. Okay. But actually, when we are going for in vivo studies, we are not going to that human trials. We will only
I understand.
focus on that.
We most probably go the musculus.
Okay. And for in vitro studies also, we will use some kind of proteins. Okay. So actually, when we are using in vitro studies, sir already told us about the Ellman method or modified Ellman method. We will purchase acetylcholinesterase enzyme and we will go for in vitro studies. Okay. But my doubt is actually when we are going for that in vitro study, whether that protein has the same sequence as that of the protein we are doing in the in silico.
Yes, it will be having 90%. It will be matching. Yesterday's Mr. Samina have discussed about homology modeling, right?
Yes.
If you are not having the protein structure, we'll be taking the strand and template. Like that, our body condition, our protein sequences are the right. The same way, some organisms will be having the sequence. So almost, I'm not saying 100% sure, but 90% it will match with the sequence of our protein structure as well as the organism which they have taken. So they have been adapted and the assays have been validated. It have been proved so that we are carrying out the assays.
Okay. Thank you, sir.
Thank you, sir.